Mechanism of hearing. To understand the mechanism of hearing, we have to know some physics related to sound. Sound is produced by vibrations transmitted in air, liquids, or solids. Sound is produced by vibrations transmitted in air, liquids, or solids. And the velocity of sound is higher in solids than liquids than air. Vocal cords vibrates during expiration by the air coming from the lungs. Vocal cords vibrates by air coming from the lungs during expiration, and this produces a sound which is transmitted to the air, Sound has a group of characteristics or two main characteristics. The first one is the frequency of the vibration, and the human ear can perceive sounds between 20 to 20,000 hertz or cycle per second. That is the normal sound frequency that we can perceive. Also, sound has something which is called intensity, and the unit of the intensity is the decibel. The unit of the intensity of the sound is known as the decibel. And the 1 decibel increase in the intensity of the sound means an increase tenfold of the sound intensity. Tenfold increase in the sound intensity. So, sound has frequency and has intensity. The mechanism of hearing, and before we are going to discuss the mechanism of hearing, we have to describe the structure of the organ that perceive the sound or transmit the vibration of the sound waves to action potential or to a nerve impulse. The ear has three principal regions. The first one is the external ear, and the external ear conduct and collect sound waves. It includes the air pinna and the external ear canal, and both of them are working to collect and conduct the sound waves, directing them to the tympanic membrane. And the tympanic membrane is a membrane that separates the external ear from the middle ear. Separates the external ear from the middle ear, and the middle ear is a air-filled cavity that contain three bony ossicles used to produce conduction and magnification of the sound waves. Conduction and magnification of the sound waves from the external ear to the inner ear. So we have three bony ossicles. These three bony ossicles are vibrating, and when they are vibrating, they transmit the sound waves from the external ear to the inner ear. If we go to the inner ear or the internal ear, it is present in the labyrinth, is known as the labyrinth. And the labyrinth is formed of group of tunnels and cavities. These group of tunnels and cavities are arranged into shapes. The first one is the cochlea, and the second one is the vestibular apparatus. The vestibular apparatus contains two cavities. One is known as the utricle, and the other one is known as the saccule, plus semicircular canals, plus three semicircular canals. This labyrinth is consisting of two parts: outer bony labyrinth. So we have this one, which is outer bony labyrinth, and inside we have inner membranous labyrinth. So the labyrinth is formed of outer bony labyrinth, inner membranous labyrinth, and this labyrinth is present in the vitreous portion of the temporal bone of the skull. It is present in the vitreous portion of the temporal bone of the skull, and as I mentioned that it is formed of the cochlea, three semicircular canal, plus two cavities. One is known as the utricle and the other one is the saccule. This labyrinth or the internal ear is going to produce what? Transformation of the impulses or the sound waves or the balance impulses to action potential. So labyrinth generates action potential to transmit sound and balance information to the brain. It is formed of bony labyrinth and membranous labyrinth, and as I mentioned, it is formed of group of tunnels and cavities arranged in the form of cochlea, which perceive the sound waves and transform the sound waves into action potential. The vestibular apparatus that's formed of utricle, saccule, and three semicircular canal that are regulating our posture and balance. The inner ear or the labyrinth. The inner ear or the labyrinth is formed of cochlea and vestibular apparatus. Cochlea, vestibular apparatus, cochlea in the form of three coiled tubes, and it has an outer portion called outer bony labyrinth, inner membranous labyrinth, and the vestibular apparatus consists of utricle, saccule, utricle, saccule, and three semicircular canals. Outer bony labyrinth, which includes the inner membranous labyrinth. So, as we mentioned, we have outer bony labyrinth, and we have inside the membranous labyrinth, the inner membranous labyrinth. In the space between the bony labyrinth and membranous labyrinth, we have a fluid which is called the perilymph. And this perilymph is similar to the extracellular fluid in concentration. It has high sodium and low potassium. While the membranous labyrinth is containing another fluid which is known as the endolymph. And this endolymph is similar to the intracellular fluid. So here we have bony labyrinth, membranous labyrinth. In between we have a fluid called the perilymph, similar to the extracellular fluid. And inside the membranous labyrinth, we have another type of fluid which is called the endolymph, and it is similar to the intracellular fluid. This means it contains high concentration of potassium and low concentration of sodium. Membranous labyrinth contains receptor for hearing in the cochlea and the receptors for the regulation of the equilibrium in the vestibular apparatus on the vestibule. Anatomy of the ear. If we look to the structure of the ear, as we mentioned, it is formed of the external ear, middle ear, and we have the inner ear. The part of the inner ear concerned with the transformation of the vibration of the sound into action potential is the cochlea. So the auditory receptors are located in the cochlea. Auricle and external ear canal are collecting and conducting the sound waves to the tympanic membrane, and the tympanic membrane separates between the external ear and the middle ear. It is separating between the external ear and the middle ear. The middle ear transmits the vibration of the tympanic membrane to a special window here called the oval window, and this oval window is going to transmit the vibration to the cochlea, and we have here a tube which is called the auditory tube, and this auditory tube, or named as Eustachian tube, it produces what? It produces equalization of the pressure on the tympanic membrane. It is not permanently opened or it is not permanently closed, but it is opened during deglutition or swallowing. It is opened to allow the pressure to be equalized across the tympanic membrane, So that is the external ear, middle ear, and the inner ear, and the organ of hearing in the inner ear is what? Is the cochlea. The three bony ossicles in the middle ear are known as malleus, incus, and the stapes, and the stapes is articulating with the oval window to transmit the vibration to the. Moving to the middle ear, air-filled cavity has two openings. One is called the round window, and the other one is called the oval window. One is called the round window, the other one is called the oval window. And the function of the middle ear is the conduction and magnification of the sound waves. As the sound waves or the vibration of the sound are transmitted from air medium or the external ear to the inner ear, from an air medium to liquid medium, most of energy is lost. So the function of the bony ossicles, is the preservation or the magnification of the sound waves and preventing it from loss. So we have the malus articulating with incus, then articulating with the stapes, and the stapes is going to transmit the vibration to the fluid which is present in the inner ear. The anterior wall of the middle ear is going to or leads to the auditory tube or the Eustachian tube, which connect the middle ear with the nasopharynx. Cochlea or the hearing apparatus. If we would like to look for the structure of the cochlea is formed of three tubes, and these three tubes are coiled to make 2.5 turns around a central bony axis. So if we have three tubes and these three tubes are coiled, and when they are coiled around a central axis, they are going to make about 2.5 turns. And if we look to the three tubes, we have one here called scala vestibuli, this is the uppermost one, scala vestibuli, and we have the scala media or the cochlear duct, and we have the scala tympani or scala tympani. So we have scala vestibuli, cochlear duct or the scala media, and we have scala tympani, which are the three tubes. So these three tubes are coiled and make 2.5 turns around the central bony axis called the modulus. We have here three tubes. Scala vestibuli and scala tympani are filled with perilymph, while the scala media or the cochlear duct is containing endolymph. So here we have three tubes. These three tubes are separated by two membranes, as we will see in the next slide. So the cochlear duct or the scala media is the continuation of the membranous labyrinth into the cochlea. It is filled with endolymph. And the channel above the scala media is the scala vestibuli containing perilymph. And the channel below or the tube below or the other one, which is scala tympani, containing also perilymph. And both of them are continuation of the space between the bony labyrinth and membranous labyrinth, and they contains perilymph. We have here a cross section in the cochlea, and we find the uppermost tube is scala vestibuli, lowermost tube is the scala tympani, and in the middle we have the cochlear duct or we have the scala media. Here we have perilymph, the space here or the scala media is containing endolymph. The three tubes are separated by two membranes. In between the scala vestibuli and scala media we have the vestibular membrane, and in between the scala media and the scala tympani we have the basilar membrane. So if we are going to produce spreading or allowing these three coiled tubes to become spreaded, we have scala vestibuli, scala tympani, both are containing perilymph, and here we have the scala media. And we have here the foot plate of the stapes, and the foot plate of the stapes that bring the sound waves from the tympanic membrane and the bony ossicles is going to direct these vibrations to the scala vestibuli. Is going to direct the vibration of the sound waves to the scala vestibuli. So again, we have a cross section in the cochlea, and this is the shape of the three tubes: scala vestibuli, scala media, and scala tympani. The membrane here is known as vestibular membrane. The membrane here is known as the basilar membrane. Over the basilar membrane, we have an important structure called organ of Corti. And this organ of Corti is containing the auditory receptors. So the organ of Corti, which is located over the basilar membrane, is containing the auditory receptors. And from the base of the auditory receptors, we have the afferent neurons of the auditory nerve. We have the afferent neurons or the sensory neurons of the auditory nerve or the cochlear nerve. Organ of Corti. What is the shape of the organ of Corti? If we look for the organ of Corti, which rests on the basilar membrane, is formed of the hair cells. We have two sets of hair cells: outer hair cells and inner hair cells. We have two sets of hair cells. We have outer group and we have inner group. And we have cilia coming or projecting from the hair cells. These hair cells are provided with stereocilia, or small hair processes coming up from the upper border of the hair cells. And these cilia are embedded in a gelatinous membrane called the tectorial membrane. So again, that is the basilar membrane, and over the basilar membrane we have the organ of Corti, and the organ of Corti is consisting or containing two sets of hair cells: outer hair cells, inner hair cells, and the upper border of the hair cells are provided with cilia called the stereocilia. And these stereocilia are projecting toward the tectorial membrane, which is a gelatinous membrane. Inner hair cells is considered or concerned with the detection of the sound waves and transmit these sound waves into action potential through the auditory nerve. So the transformation of the sound waves is a function of the inner hair cells, while the outer hair cells perform an amplifying role through stretching of the tectorial membrane. What is the mechanism of hearing? The mechanism of hearing include the following steps. Number 1, collection and conduction of the sound waves by the external ear. Then, vibration of the tympanic membrane, conducting this vibration to the inner ear through the bony ossicles or across the bony ossicles or by the bony ossicles. Then the foot plate of the stapes is going to produce pushing or inward movement against the oval window, and this will allow the vibration to be transmitted to the scala vestibuli. When you produce vibration of the scala vestibuli, you are producing vibration of the vestibular membrane, transmitting the vibration to the endolymph. And when you transmit the vibration to the endolymph, you are going to produce vibration of the basilar membrane. So when the sound waves vibrates the perilymph in the scala vestibuli, is going to produce vibration of the vestibular membrane, transmitting this vibration to the endolymph in the scala media, vibrating the basilar membrane. So when you vibrate the basilar membrane, when you vibrate the basilar membrane, you are going to produce upward and downward movement of the basilar membrane. So you are going to push the hair cells and the stereocilia against the tectorial membrane. Again, vibrating of the basilar membrane up and down, causing the hair cells to strike against the tectorial membrane. So the cilia is going to show bending. The cilia is going to show bending against the tectorial membrane. The bending of the cilia opens group of potassium channels, groups of potassium channels producing potassium influx, and the production or the entry of potassium to inside the cell produce depolarization. So here the mechanism of depolarization is by potassium influx, and this will produce what? Change of the release of the neurotransmitter, change of the neurotransmitters coming from what? From the base of the hair cells to generate action potential in the auditory nerve, to generate action potential in the cochlear nerve or in the auditory nerve. So again, what are the steps of the mechanism of hearing? As we discussed previously, we have conduction of the sound waves to the tympanic membrane, then the vibration transmitted through the bony ossicles to the foot plate of the stapes against the oval window, then vibrating the fluid here, which is perilymph, vibrating the vestibular membrane, then transmitting the vibration to the endolymph, transmitting the vibration to the basilar membrane. When the basilar membrane vibrates, it moves the organ of Corti up and down, so the stereocilia are striked or moved against the tectorial membrane, opens potassium channels, potassium influx, depolarization of the hair cells, change the release of the neurotransmitter. That's one of the hair cells under the electron microscope. And when they are stimulated by depolarization, they release a neurotransmitter stimulating the afferent neurons of the cochlear nerve and generating action potential. What is the function of the round window? What is the function of the round window? The round window, which is bringing again the vibration or the excess vibration, bringing it back to the middle ear. So it dissipates the excess pressure or acts as a relief hole. A release hole or a relief hole, allowing the excess pressure to be released to the middle ear. So the round window acts as a relief hole or dissipates the pressure from the cochlea back to the middle ear. That is the function of the round window. It dissipates the excess pressure. How can we differentiate between sounds regarding or according to frequency or the determination of the sound frequency? We have here the basilar membrane, that is the base of the cochlea, that is the apex of the cochlea, and the basilar membrane, when they studied, it is formed of multiple fibers, and these fibers are arranged from the base of the cochlea to the apex of the cochlea. And they found that the high frequency sound producing vibration of the basilar membrane mostly at the base of the cochlea, So when you listen or hear a high frequency sound, mostly the vibration will occur in the basilar membrane at the base of the cochlea. So most of the hair cells are stimulated in this region, and they are going to send impulses. These impulses are carried to the brain, to the primary auditory area, which have a special map for the sound frequency. So when the base of the cochlea or the basilar membrane at the base of the cochlea vibrates more, the hair cells at this region going to send the impulses to the primary auditory area, and the primary auditory area will identify the sound frequency according to the stimulated hair cells. While the low frequency sounds produce more vibration to the basilar membrane at the apex of the cochlea, produce more vibration at the apex of the cochlea. So when the hair cells are stimulated in this region, so they are going to send the impulses to the brain or to the primary auditory area, and this will allow to stimulate some neurons in the primary auditory area, informing that this sound is a low frequency sound. So we can consider the basilar membrane as a frequency analyzer. We can consider the basilar membrane as a frequency analyzer. So, what is the function of the basilar membrane? It acts as a frequency analyzer. High frequency sound vibrates the basilar membrane most at the base of the cochlea, stimulating the hair cells at this region, sending impulses to the brain, identifying that the sound is high frequency. Low frequency at the apex of the cochlea, stimulating of these hair cells, giving impulses that the sound is of a low frequency criteria. How can we determine the intensity of the sound? High intensity sound waves cause large vibration, and this will increase the rate of nerve impulse generation and stimulating a large number of hair cells. So we are going to identify the intensity of the sound by what? By the rate of nerve impulse generated and also by the number of stimulated hair cells. High intensity sound will increase the number of impulses and the number of stimulated hair cells. Auditory pathway. How can the signals are transmitted to the primary auditory area in the cerebral cortex? We have here the afferent neurons of the auditory nerve or of the cochlear nerve, and that is the impulses will be generated here in the cochlea, transmitted across the cochlear nerve to the cochlear nuclei. And when the cochlear nuclei stimulated, they are going to send the impulses on the same side and on the opposite side. So the impulses or the second order neuron arising from the cochlear nucleus will ascend on the same side and the opposite side to relay in the superior olivary nucleus. And from the superior olivary nucleus, we have impulses that will ascend through the lateral lemniscus to the inferior colliculus in the midbrain. So from the cochlear nucleus to the superior olivary nucleus on both sides and to the inferior colliculus of the midbrain to the inferior colliculus in the midbrain on both sides, and from the inferior colliculus the fibers will go to the medial geniculate nucleus in the thalamus, and from the medial geniculate nucleus the impulses will project to the primary auditory area in the temporal lobe to the primary auditory area in the temporal lobe. So from the hair cells to the cochlear nucleus to the superior olivary nucleus on both sides to the inferior colliculus and then to the medial geniculate nucleus in the thalamus to the cerebral cortex to the primary auditory area on both sides. So the auditory signals or the impulses of hearing will be carried to the cerebral cortex on both sides. Each region of the basilar membrane is linked to a specific area of the primary auditory cortex. Deafness is a significant decrease or loss of hearing. It is a significant diminution or loss of hearing. And deafness is classified into two categories according to the etiology. If we have a problem in the conduction or a defect in the conduction in the external ear or in the middle ear, this is known as conductive deafness. If we have a defect in the inner ear or in the afferent pathway or in the sensory pathway of hearing, this is going to produce sensory neural deafness. So we have conductive deafness or sensory neural deafness. Conductive deafness due to defect in the external ear by the presence of foreign body or the accumulation of wax or perforation of the tympanic membrane or the presence of infection in the middle ear is going to produce conductive deafness. Damage of the hair cells as by intake of drugs or by the prolonged exposure to high intensity sound, this can produce sensory neural deafness. Physiology of equilibrium. The physiology of equilibrium is produced by the vestibular apparatus located in the inner ear. That is the shape of the vestibular apparatus. It is formed of two cavities, utricle and saccule, and the semicircular canal. And as any part of the labyrinth, there is bony labyrinth and membranous labyrinth. The part between or the space between bony labyrinth and membranous labyrinth contain perilymph. Inside the membranous labyrinth, we have the endolymph. There are two kinds of equilibrium balance. We have the static equilibrium or the maintenance of the position of the body, mainly the head, relative to the force of gravity. So we have receptors confined or produce regulation of this or can detect the position of the head in relation to the gravitational force, and this is known as the static equilibrium. And we have the dynamic one, the dynamic equilibrium, and is the maintenance of the body position or the regulation of the body position in response to sudden movements as occurs with linear acceleration or the angular acceleration and deceleration. So with the acceleration and deceleration, either it is linear or angular, we have specific receptors to produce or to perceive these types of movements in order to maintain our equilibrium or our balance. Static equilibrium. The detection of the head position in relation to gravity is mainly produced by the receptor, which is the macula located in the utricle and saccule. The macula located in the utricle and saccule. The macula is in the shape of elevated ridge of epithelial cells, that is the shape of the hair cells or the epithelial cells. Upper border is provided with one thick large motile called kinocilium and small short motile called the sterocilium. And that is the shape of the hair cells, and they are covered with a gelatinous mass called the copula, and this copula contains the calcium carbonate crystals that form the otolith membrane. The macula consists of hair cells, and the hair cells has many sterocilia and only one kinocilium. The gelatinous or the otolith membrane that is present in the copula can produce change in the orientation of this cilia. The bending of the hair cilia or the hair cells can either be towards the kinocilium or away from the kinocilium to produce either depolarization or hyperpolarization. Bending of the sterocilia towards the kinocilium produce depolarization, away from the kinocilium produce hyperpolarization. So when your head is present in the upright position, you have something which is called basal symmetrical charge. That is the afferent neurons of the vestibular nerve. When you have the presence or you are present in the upright position, you have resting membrane potential of both cells, and the presence of the resting membrane potential maintains the basal symmetrical charge from both cells. Static equilibrium or detection of the head position in relation to the gravitational force. An otolith membrane and the otolith respond to gravity when head position is changed. When we have tilting of the head, the force of gravity move the otolith membrane and the calcium carbonate crystals. This will produce change in the orientation of the sterocilia from the kinocilium to produce bending of the sterocilia away from the kinocilium or towards the kinocilium. This will change the resting membrane potential of the hair cells to produce what either depolarization in some cells or hyperpolarization in the others, and this will produce what asymmetrical charge. This asymmetrical charge will inform our brain that we have been exposed to what, to a change of the head position. Dynamic equilibrium. And we have here example of the dynamic equilibrium that occurs in the cresta. You can understand it more from the video I'm going to put with the lecture. The equilibrium pathway. The equilibrium pathway is the pathway that's arising from the vestibular receptors, macula and the cresta, and these neurons coming from the macula and the cresta are going to relay in the vestibular nuclei, located in the medulla oblongata and bones. From the vestibular nucleus, we have impulses that's going to affect what? Affect the muscles of the body through the vestibulospinal tract. There is a descending tract coming from the vestibular nucleus, go downward in the spinal cord to affect the position of the skeletal muscle or the activity of the skeletal muscles. Also, some of the impulses go to the cerebellum. Some of the fibers coming from the vestibular nucleus go to the cerebellum. And we have pathway coming from the vestibular nucleus going to affect the accessory nucleus which detect the position of the muscles of the head and the neck, that is supply the muscles of the head and the neck. From the vestibular nucleus we have another impulses going to the oculomotor, trochlear and abducent nuclei. These nuclei are controlling the muscles of the eye, Also, from the vestibular nucleus, we have impulses going to the thalamus, and from the thalamus going to the vestibular area in the cerebral cortex, to the vestibular nucleus in the cerebral cortex. The vestibular nuclei also receives input from the eyes and from the proprioceptors, So again, the vestibular nerve relay in the vestibular nucleus. From the vestibular nucleus, impulses go to inform the cerebellum, go to the spinal cord to affect the state of our skeletal muscles by the vestibulospinal tract, to the accessory nucleus that is affect the muscles of the head and the neck, to the oculomotor, trochlear and abducent nuclei on both sides to affect the position of the muscles of controlling the muscles of the eye. And we have also impulses going to the thalamus and to the cerebral cortex. Olfaction and gustation. Both of them are considered as chemoceptive sensation and exteroceptive sensation. We perceive the chemicals present in the odors or present in the odorant molecules by smell, and we perceive the taste and or the chemicals that give the taste of food by the taste receptors present in the taste buds of the tongue. Both of these sensations are linked to the hypothalamus and limbic system. So they go to the cerebral cortex to inform the cerebral cortex, and plus the cerebral cortex, they send some of the collaterals or the information to the limbic system and the hypothalamus to evoke or produce the emotional response to food. And usually the process of taste and smell are linked together. In the last year, there is an article that is give a new discovery about the presence of smell receptors in the tongue, the presence of olfactory receptors of the tongue. So the olfactory receptor is not present only in the olfactory epithelium in the roof of the nose. It is also some of the olfactory receptors have been discovered in the tongue or in the taste buds of the tongue. And usually, if you have a common cold or a flu, what's happened? You lose the taste of food. You have a problem in the nose, but the problem also affecting the taste of food. So there is a connection between these types of sensation. Why? Because usually they regulate your food intake. They regulate the function of the gastrointestinal tract. They regulate the salivary secretion. They regulate the conditioned reflexes that produce what secretion of the gastrointestinal enzymes, and also regulate the motility of the gastrointestinal tract. So both of these sensations are controlling a lot of function in the gastrointestinal system. Olfaction is produced by the olfactory epithelium. And this olfactory epithelium is present in the roof of the nose, and the superior nasal concha. We have the olfactory epithelium that contains the olfactory receptors. It is present in the roof of the nose and the superior nasal concha. And this olfactory epithelium consists of three types of cells. The first is the olfactory receptors. That is the shape of the olfactory receptors. They are bipolar neurons bipolar neurons with cilia called the olfactory hair. With cilia called the olfactory hair. This is the bipolar neurons, and they are provided with cilia called the olfactory hair. And they have the capacity to bind with the odorant molecules. They have the capacity to bind with the odorant molecules. There are 10 to 100 million of these receptors in the nose. Then we have supporting cells to provide support and nutrition. And we have basal cells. Basal cells, they are considered as stem cells to produce regeneration of the olfactory receptors. And they usually regenerates within 60 days. The duration needed for the regeneration of the olfactory receptors is about 60 days. So the olfactory epithelium is a part that perceives the odors. It is present in the roof of the nose and superior nasal concha. It's apparently yellowish to brown in color, and its surface area is 5 square centimeters, consists of 3 types of cells: the olfactory receptors, which is bipolar neurons, and this bipolar neurons has cilia called the olfactory hairs that bind with the odorant molecules. We have supporting cells and basal cells. The olfactory apparatus can detect about 10,000 different odors, often in a concentration as low 1 over 25 billions of milligram per millimeter of air. So they have the capacity to detect different types of odor whenever the concentration of them is very low. The mechanism of stimulation of the olfactory receptors is dependent on the presence of olfactory receptor protein, or odorant binding protein. This odorant binding protein bind with the odorant molecule, and this odorant molecule should be volatile, should be water soluble, should be lipid soluble. So a group of criteria should be available to allow this odorant substance to be detected. Number 1, volatile. Number 2, water soluble. Number 3, lipid soluble. This odorant molecule is going to bind with the odorant binding protein, and this will produce activation of G protein, and this G protein is going to activate the adenyl cyclase enzyme to hydrolyze the ATP into cyclic AMP, and cyclic AMP is going to trigger or activate sodium channels, allowing sodium influx and depolarization of the olfactory receptor membrane. Depolarization of the olfactory receptor membrane. So the odorant substance should be volatile, water soluble, and lipid soluble, bind with the odorant binding protein, activating G protein receptors or activating G protein molecule that's going to produce what? The activation or the formation of cyclic AMP, which opens sodium channels, producing depolarization of the olfactory receptors. Then the impulses is going to be conducted to the cerebral cortex or the olfactory pathway. After stimulation of the olfactory receptors by the odorant substance with depolarization, this action potential is going to be generated and conducted to the olfactory bulb. That is the olfactory bulb, and it consists of olfactory glomeruli, and these olfactory glomeruli contains group of cells called mitral cells and tufted cells, and then the impulses is going to be conducted to the higher cortical areas, mainly to the primary olfactory cortex in the temporal lobe, and also to the secondary olfactory cortex Once generated, the nerve impulses travel through the olfactory nerve to the olfactory bulb to the olfactory tract directly to the primary olfactory area of the cerebral cortex. The olfaction is a sensation that goes directly to the cerebral cortex without relay in the thalamus. So olfaction is a sensation that goes directly to the cerebral cortex without relay in the thalamus. Olfaction is considered as a rapidly adapting sensation, as there is complete accommodation to the odor within 1 to 2 minutes. There is rapid adaptation to the odors within 1 to 2 minutes. Sense of taste or gustation. The gustation or the sense of taste is for detecting 5 primary tastes. 5 primary tastes can be distinguished by the taste buds located in the tongue. We have the ability to detect sour, sweet, bitter, salty, and umami. These are the 5 tastes that can be distinguished by the taste buds. Our taste buds are formed of 3 types of cells. We have the gustatory receptor cells that are provided with gustatory hair cells, and they have the ability to detect and to bind with the tastent molecules, supporting cells and basal cells. And if we look to these taste buds are found in the papilla of the tongue. So the receptors for sensation are located in the taste buds found in the papilla of the tongue. There are about 10,000 taste buds that are mainly found on the tongue, but they are also distributed in the soft palate, pharynx, and epiglottis. They are also distributed in other parts like soft palate, pharynx, and epiglottis. The mechanism of stimulation of the taste receptors are different. For example, the salt receptors are stimulated by sodium influx. Salt receptors that detect the sodium chloride present in food are stimulated by sodium influx. The sour receptors are stimulated by hydrogen influx, while sweet and bitter receptors are stimulated through G protein coupled receptors with the generation of cyclic AMP that produce closure of potassium channels. Umami depends on the detection of glutamate that can bind to G protein coupled receptors and stimulate or producing depolarization of the receptors. Depolarization of the receptors by increasing the release of calcium from the endoplasmic reticulum by the presence of diacylglycerol inside the receptors. So its depolarization is dependent on G protein coupled receptors. So at the end, there is depolarization of salt, sour or sweet, bitter, and umami. But how can our brain identify the depolarization is caused by salt or depolarization caused by the presence of sour or the presence of sweet? This can be differentiated by the neurotransmitter in the sensory pathway. Each type of receptor has a specific pathway, and also each type of receptor releases a specific neurotransmitter in its pathway to allow the brain to identify and differentiate between the different types of taste. It is well known that the sweet or the sugary foods increase the dopamine in your brain. So the neurotransmitter that's released in the brain in response to the presence of sugars or presence of sweet in your mouth or in your food is dopamine. So when the dopamine is released in your brain, it gives you the sensation of the sweet or the sugary food. The gustatory pathway. The sensory impulses or the impulses generated by stimulation of the taste buds are carried through the facial nerve from the anterior 2/3 of the tongue, from the glossopharyngeal nerve or through the glossopharyngeal nerve from the posterior third of the tongue, and from the throat and epiglottis by the vagus nerve. And all these signals are conducted to the gustatory nucleus in the medulla oblongata. And from the gustatory nucleus, the impulses are going to the thalamus. And then the third order neuron coming from the thalamus projected to the primary gustatory area of the cerebral cortex on the same side. And some of the impulses will project to the hypothalamus and limbic system. Some of the fibers from the medulla is going to project to the hypothalamus and limbic system to produce the emotional behavior related to food intake. The emotional behavior related to food intake. Taste signals from the thalamus project to the primary gustatory area of the cerebral cortex on the same side to give the conscious perception of taste. So that is the taste pathway from the anterior two-thirds of the tongue through the facial nerve, from the posterior third of the tongue through the glossopharyngeal nerve, from the tonsils and from the epiglottis or the throat through the vagus nerve to the gustatory nucleus in the medulla oblongata, then to the thalamus, to the primary gustatory area of the cerebral cortex. Some fibers go to the hypothalamus and the limbic system to control the emotional behavior related to food or related to the taste of food.